Hello, everybody, and welcome. I'll be the chair for this session. So I'll tell you a little bit how we do work now. He will give us a presentation, his thesis titled An Acetogen-Based Renewable Chemistry. He was supervised in his PhD by Alex Nielsen. And on the other hand, we have our PhD committee that will be examining this PhD defense. We have Kongens Lyngby, associate professor at the Biosustainability. We have Simon Niels who's assistant professor at the University of California, Riverside. We have Alfred M, who is associate professor at the University of Tartu in Italy.
Thank you so much for the introduction, Stefano. I'll be presenting my PhD work. So this is 4 years of work. So as Stefano said, my thesis is called An Acetogen-Based Renewable Chemistry. And this theme is kind of rooted in our modern day dependence and use of fossil resources. Fossil resources are very fundamental to our modern day society, and we use huge amounts of them for energy purposes. So we burn them for fuel, heat, and power, but we also use them for the production or for the feedstock of our petrochemical industry. And just to visualize how much of these resources we actually use, the cover of the magazine I presented here to the left is a good illustration I found of how much of the 3 types of fossil resources we extract and use every single year. So the building, the tiny building you see in between there is the Burj Khalifa, which is for the moment the tallest building in the world. So yeah, each single cube is more than twice the height of the tallest building in the world. So there are a few consequences to the use of these amounts of fossil resources, and that's firstly that it's a depletion of finite resources, so they're not endless. But more importantly, and we hear increasingly much about this, is that they emit a lot of greenhouse gases, especially CO2. So the combustion of these will end up in the atmosphere where it warms up the planet and increases the global temperature. So yeah, the aim of this thesis is to tackle the chemical industry, so the petrochemical industry. And just to give a really brief overview of how the petrochemical industry looks, it's generally a very linear process where we go from extraction to the left to the consumables that we all are very accustomed to, which tend to have a very short lifespan. These then get discarded and trashed. So after this very short lifespan, and then in general, they get incinerated. So the carbon that's in there will end up in the atmosphere. There's a lot of claims in especially plastic industry about recycling of plastics. But these are very misleading because it's only marginal amounts that are actually being recycled. And it's not really recycled, it's rather downcycled. So overall, it's a very linear industry going from the feedstocks to the combustion. Increasingly in modern dialogue, we hear a lot about the ongoing shift to renewable energies, but we barely hear anything about an ongoing shift towards renewable chemistry. So that's what we're going to talk about in this thesis. And the proposed solution here is the use of CO2 as a sustainable feedstock. So the end product of this linear extraction and use to recircularize it to produce new things out of it. And this is done by gas fermentation using microbes. These microbes, they produce, so they're called acetogens, hence the title, and they produce acetate and ethanol as intermediates. So the goal is not per se to replace the whole petrochemical industry with these bacteria, but rather to replace the feedstock that they use. So that acetate and ethanol are intermediates which could be converted to the large variety of compounds we are used to nowadays. So what are these acetogenic bacteria? So to define them in 3 words, they are orthotrophic, lithotrophic, and anaerobic. And by that, I mean that they're orthotrophic, that they're able to grow on inorganic carbon, in this case CO2, as opposed to many other organisms like ourselves, who would rely on sugars, etc. So organic carbons. They are able to use this inorganic carbon, so this CO2, based on energy that they find from chemicals, most notably hydrogen. Because also the use of CO2 is not that special. Plants do it too, but plants use the sunlight as energy. And these bacteria, they are able to use CO2 and hydrogen to produce acetate, but also to replicate, so to form more biomass. And to do so, they use the William-Demant pathway. And just to give a brief overview of this reaction, to the left here, we see two big molecules of CO2, which will react with 4 molecules of hydrogen, dihydrogen, to produce acetate to the right and water as a rest product. And this reaction generates energy, which the cells can use to grow. And the reason I show this is to demonstrate that to the left, we start with the lowest energy form of carbon. So this completely oxidized carbon, so CO2, so the oxygens are there. And this is so abundant and very useless for cells, etc., that you need to input energy to get useful molecules, such as acetate. So that's why this is an interesting reaction, because it goes from inorganic to organic. And it's so it's a reduction of oxygenated compounds. And how this William-Demant pathway looks, so the pathway that catalyzes this reaction would be as such, where we see at the top, we start with the CO2, and this CO2 gets stepwise reduced to the more organic matter. So the CO2 gets converted to formate first, and then this formate will bind to a cofactor. And this cofactor is called tetrahydrofolate, or H4F here. And it will look a bit like this. So this is the chemical structure, where the large molecule in black is the tetrahydrofolate, and the small molecule in red would be this formate. And the formate will then bind to the cofactor, where in a stepwise way, with the oxygens would be replaced by hydrogens. So this is the reduction of this CO2. And then you end up with the CH3 group, the reduced variants, which is useful for the cells to utilize. And then this is kind of where the magic happens, so to say, where it will take this reduced CH3 group, and then with another group of enzymes, it will combine it with a carbon monoxide, so a CO, and this will link two 1-carbon compounds together to form a 2-carbon compound, namely acetyl-CoA. And that would look like this, where we have the tail to the left being the CH3, the reduced step, and the carbon monoxide here bound to this cofactor called coenzyme A. And the tetrahydrofolate cofactor I was describing earlier gets discarded again. Now, from this acetyl-CoA is an extremely versatile compound, which is at the core of cellular metabolism in basically every organism. And the cells, they can go, the metabolism can go one of two ways. Either it will create biomass from acetyl-CoA, which is the whole variety of compounds that one would imagine forming a cell. Or it would form acetates, where this coenzyme A would get reacted away, and this reaction generates energy, which restores energy that is needed for the reduction. So that is how they produce energy, and the biomass is how they reproduce. Now, with this background given, I would like to give a brief overview of the chapters included in my thesis, and also the types of problems that I'm addressing in these chapters. So the first chapter is called Acetogenesis as a sustainable alternative to petrochemistry. And in this chapter, we have a policy type research question, which can also be called a wicked research question, because it's highly interdependent and complex. And it is a type of problem that we're maybe not so familiar with in a technical university. But it basically has no true or false answers, just good or better answers as such. And the hypothesis, the research question was formulated as, can acetogenesis form the basis of a sustainable chemical industry? Then the second chapter is a more engineering type research question. And the research question is, what are the optimal conditions for acetogenic fermentation, and what could be produced? So it's kind of a two-part research question. And then lastly, The last chapter is a very fundamental research question. So this is old school science, so to say. And the title of the chapter is Methylotrophic Acetyl-CoA formation as a pathway at the origin of life, where the scientific research question is, what is the evolutionary history of the Wood-Ljungdahl pathway? So the pathway that does the CO2 reduction. So to start with the first chapter, Acetogenesis as a sustainable alternative to petrochemistry. So again, the research question is, can acetogenesis form the basis of a sustainable chemical industry? And I was saying that these wicked research questions are highly complex and interdependent. So one way of addressing them is to divide them up in sub-research questions. So that's how we address this, this chapter. So the first sub-research question in that regard is to define what the current anthropogenic climate impact is. And to start this off, we use data from the International Panel for Climate Change, which we visualize to the left here, which quantifies the amounts of carbon stored in different geological or stocks on the globe. And what you see is, for example, here to the bottom is the fossil resources, and I've indicated the depletion by the lighting part in the pie chart. And the increase, so depletion and increase are compared to pre-industrial times, so roughly 200 years ago. And the increase is shown in the red circles. So that shows how much of the greater circle has been added by human activity. So yes, we see fossil has been almost depleted by half, which are quite staggering numbers. And most of this, so when fossil resources get depleted, they get burned, and the CO2 that's in there will end up in the atmosphere. And that's where you see the largest increase, where we see now, compared to 200 years ago, a plus 47% increase of carbon in the atmosphere. So quite gigantic changes. And from the atmosphere, part of it can be dissolved in the oceans, because CO2 is actually quite soluble. So that's why we see that the ocean is actually the largest carbon stock. It didn't even fit in the schematic. So there we see a very small percentual change, but the ocean is clearly a gigantic carbon stock. And the fact that it's a small percentual change doesn't mean it's harmless, because there are still major consequences to the carbon in there, which primarily is the acidification of oceans and the consequences that follow with that. So to put this is just to put into perspective the human impact so far. Then the next question is, how is the petrochemistry's contribution to this greater picture? Because the petrochemistry is what we aim at replacing. And to identify this, we first looked at the types of emissions. So there's energy types of emissions, which are basically just the combustion or the burning of these fossil resources, where the carbon that's in these resources will just end up in the atmosphere. But then that's not the only type of emissions of greenhouse gases that there is, and CO2 is also not the only greenhouse gas. There's also direct emissions, and direct emissions would be emissions that are not per se from combustion, but can be, for example, methane emissions from cattle, which is a big contributor. It could be the decomposition of waste. So wastewater treatment releases a lot of CO2, or landfills do that too. There's also the cement industry, which releases a lot of CO2, etc. There's several others. And then the question is, how does this pie chart, how does it distribute between sectors? So which sector is actually responsible for these numbers here to the bottom? So overall, we see that there is around 50 gigatons of CO2 equivalents per year being emitted. So how this links has been represented here in the Sankey diagram, or in the alluvial plot, how it's also called, where the values to the left are the same as in the pie charts, kind of rolled out, and then they're connected to the right side to the industries that are responsible for it. And we see some really big contributors, such as agriculture and manufacturing, both accounting for a bit more than 20%. And agriculture is for a big part is this methane from cattle. But then there's also transport, so that could be road transport, but also planes or shipping. A lot of CO2 emissions from residential and commercial buildings, so that could be heating, air conditioning, electronics, etc. But of course, the one we're most interested in is the manufacturing of chemicals, the third from the bottom. And this manufacturing of chemicals accounts for 6% of global greenhouse gas emissions, which is a very significant amount. So the final question to this is, now we know what the total impact of the petrochemistry is and what the global human impact is, but the follow-up question is then, how is this impact distributed within petrochemistry? Which processes are really accountable for these emissions? And to get a clear picture of this, we need to understand what the petrochemical industry primarily is. And it all starts with these three fossil resources, right? Coal, gas, and oil. And these three resources, they are converted to two petrochemical feedstocks, namely syngas, so it's a very energy-rich, carbon-rich gas, or naphtha, and naphtha is a fraction of oil. And the syngas can be converted to methanol and hydrogen, where the hydrogen is just taken from the syngas and all the carbon is released. And this hydrogen can then be converted to ammonia. And ammonia is the primary chemical, the largest production. And then from this naphtha, the naphtha can be put into a so-called steam cracker, and the steam cracker will produce a variety of products, which fall into four categories, which are ethylene, propylene, a so-called C4 stream, so that means that it's compounds with 4 carbon backbones, and abbreviated BTX, so it's toluene and other aromatics, so these are carbon rings. And these together are called olefins. And yes, so if you quantify how much of each of these products is produced, so ammonia is by far the most produced with 230 megatons per year, followed by methanol. And so ammonia is mostly used as a fertilizer, followed by methanol. And then the olefins, the ethylene, propylene, etc., they're produced a bit less, but still in significant amounts. And the reason that all of these products are so polluting is because they're, of course, based on fossil resources, the coal, gas, and oil, so it's a linear extraction, but also their processing relies on extremely energy-intensive steps, especially the gasification of coal is very energy-intensive. The Haber-Bosch process and the steam cracking are just require a lot of energy inputs to transform these compounds. And that's why it's a very polluting industry. Given this picture of the current petrochemical industry, the question arises, would acetogenesis provide a sustainable solution to the use of fossil resources? And to investigate this, we envisioned an alternative flowchart of how this industry would look. And to start with, here is the list of the acetogenic process, where acetogenesis can use either hydrogen and CO2 as two feedstocks to produce acetate and ethanol, or it can also produce it directly from syngas. And as you might recall, both hydrogen and syngas are products that are now already important in the petrochemical industry, but they can also be produced from renewable resources instead of fossil resources. So hydrogen can be produced from water electrolysis, which is also a very energy-intensive step, but it can be done with green renewable energy. And then syngas can be produced not from coal, but from dry organic waste materials, which would make it a circular process. And now this acetate and ethanol can be converted in turn to compounds such as these olefins, so ethylene or C4 compounds and others. Then also, besides dry organic residue as a feedstock, you could also use wet organic residue, which is already quite heavily used to form biogas. So that's methane primarily. And this methane or biogas can be then used to produce syngas, which also feeds to acetate and ethanol through acetogenesis. And then ammonia and methanol can also be produced in this greater picture, but they also rely on hydrogen. So overall, we see that hydrogen is extremely central in this alternative chemical industry. Yes, and there's no alternatives to ammonia production and methanol production than using hydrogen as a feedstock, while there is an alternative for acetate and ethanol production through acetogenesis. So it does not rely on hydrogen per se. You can also do acetogenesis from syngas. And since electrolysis is by far the most energy consuming step in this whole process, it would make sense to replace primarily or prioritize the replacements of ammonia production and methanol production, which are key chemicals to be produced, and focus acetate and ethanol production from syngas. So in conclusion, acetogenesis is a sustainable alternative to olefin production, so the steam cracker products, under given conditions, which are that it replaces the current market instead of just creating a new market on top of that, because then the emissions would not be cut. Under the second condition that it uses biomass-derived syngas as substrates, so not using this hydrogen. And then lastly, that it produces high-value compounds with a long lifespan. And by that, I mean primarily not biofuels, because putting in all this energy to produce these compounds just to combust them later on is not a logical way of proceeding. And it does not add up. Now, for the second chapter, so we have kind of shown the relevance of the acetogenic industry, or the acetogenic role in the new alternative industry. Now, the second chapter is an engineering chapter where we optimize and probe efficient microbial CO2 utilization processes. To do this, we used metabolic modeling and process modeling. So again, the research question is, what are the optimal conditions for acetogenic gas fermentation, and what could be produced? And the whole reason of writing this chapter is rooted in our research group working a lot with thermophiles. And we do this because we believe there are certain advantages to the use of thermophiles. And by thermophiles, I mean microbes that grow at higher temperatures. So think of 60 degrees. And the advantages are that there are lower cooling costs because you operate at higher temperatures. There's also a lower contamination risk, simply because most contaminants would be kind of room temperature or a bit above. So the contaminants would not be able to grow at these high temperatures. And then lastly, we think that it would allow for a continuous recovery of volatile products. So the aim is to produce compounds that would just evaporate, and then we could just take them off from the off-gas. And that would allow for an easy extraction, but also it would limit the product inhibition. So if there's a lot of products in the reactor, the cells will not want to produce more. And you avoid this by going up in temperature. But of course, we're scientists, so we don't really appreciate beliefs. And we put these theories to the test. And that's what we did in this chapter. And to start with, we investigated the gas uptake rates. And the gas uptake rate is just, so the substrate is in the gas bubbles, right? So the hydrogen and CO2 are being bubbled through this tall column. And then the gas has to diffuse into the liquid. And there's one way, a good way of modeling this, which is called two-film mass transfer model. And it just uses this equation here shown. And yes, so this equation, just to break it down, it is composed of several factors. So KLA is a mass transfer coefficient, and this mass transfer coefficient is reactor specific, and it is also compound specific. But it is also on top of that temperature dependent, which is the interesting part. And then the second term is the saturation concentration, which is not reactor dependent, but is compound specific, and it's also temperature dependent. Then there is the concentration in the liquid, which we assume is zero because the cells would just uptake everything to feed on it. And then lastly, this gives us the mass transfer rate. And people in the gas fermentation field will often tell us that why would you work with thermophilic acetogens? Because the solubility of gases goes down with temperature. And that's indeed true for the saturation concentration, C star. But it is not true for the mass transfer coefficients. And here it has been plotted as on a logarithmic scale as a function of temperature. So the horizontal bar is ranging from 0 to 80, and it will predict what this coefficient is for the mass transfer rate. And as you see, it goes up exponentially with temperature. On the other hand, as kind of well known, the saturation concentrations, they drop with temperature. And they drop kind of linearly while the KLa increases exponentially. So the question is, how does this interplay in the overall reaction? And when we put this all together in this above equation, then we see here to the right for most compounds that the overall mass transfer rate will go up with temperature. So it is beneficial for the exchange rates to go up in temperature, except for CO2, the one which is completely at the top. But that one is so much higher that it would not be limiting anyways. And CO2 is not the valuable feedstock, it would be hydrogen. And hydrogen is the deep, like the dark blue one, and that one increases the most. So gas fermentation definitely benefits of thermophilic environments. Now, besides temperature effects on the process, there is also temperature effects on the bacteria. And to investigate this, we made a black box metabolic model. And this sounds very fancy, but it's in essence, it's quite simple, where we just look at the bacteria only as its inputs and outputs. And that's represented here to the left in the figure, where we'll take up CO2 and hydrogen to produce acetate. And that reaction generates energy, which the cell needs to produce more cells. And these two reactions, they're called catabolism and anabolism. And yeah, I guess people that go to the gym are maybe familiar with these terms, and it's the same concept. So catabolism is the generation of energy, and anabolism is the generation of more biomass. And when you combine these two together, by balancing out the energy produced by the catabolism to the energy required by anabolism, you get an overall growth reaction, where you have these substrates to the left, so CO2, hydrogen, and ammonia, because the cells also need nitrogen, which produces biomass and acetate and water. And this can then be combined with, so this is just for the cells to grow, but this is not the only reactions that are going on, because besides growing, the cells will also need to spend energy just to stay alive for maintenance. And for maintenance, they will use the same catabolic reaction using CO2 and hydrogen to produce acetate. And what we saw from literature, and we modeled this, is that the maintenance energy requirement is temperature dependent. So we will go up exponentially with temperature, following this equation shown here, the Arrhenius equation. So in essence, if you combine the overall growth reaction with the maintenance reaction, then you get the real overall reaction. And then we'll look that overall reaction will allow you to predict the ratio of products to biomass. So for a certain amount of biomass, how much product is produced. And this, since the maintenance requirement is temperature dependent, this ratio is also temperature dependent. And what we see in the plot to the right is that the ratio will increase in favor of acetate production with temperature. So since we in the field of acetogenesis are not, let's say, interested in producing biomass, that's just our catalyst, we want to produce the products. That means that if you go up in temperature, you waste less of the substrates for biomass and you focus more on acetate production. Now, this was only investigating acetate production, but these acetogens, they can produce more than only acetates. I also mentioned ethanol earlier, but there's actually a wider range of products, including, so there's a lot of efforts to produce acetone from acetogenesis. And these are then pathways that needs to be added to the pathway to the organism. They don't produce it naturally, but it can be done. And they also, some acetogens tend to produce butyrate, butanol, and butanediol. And to investigate the possibility of producing these different products, And hoping, of course, to produce a volatile product, but also to produce a product that's a bit of higher value than acetate and ethanol. We made a stoichiometric model. And the stoichiometric model was able to assess the energy produced by the formation of each of these products. And it predicted that from hydrogen as a substrate, as an energy source, only acetate and ethanol really make sense and generate energy. The others would be much lower. However, instead of using hydrogen, you can also feed carbon monoxide, which is a richer substrate. And in this case, the metabolism has much more possibilities to produce more complex compounds. So it is possible to produce compounds that would be volatile, but would these products then actually evaporate? That is the next question. And previously, we've kind of worked on the assumption, looking at acetone mainly, that the boiling point of acetone is at 58 degrees Celsius. So if we would operate at 60 degrees, the reactor, the acetone would just boil off. However, there's a few problems with this. The first one being that the types of reactors acetogenesis would appear in are very tall columns. And what happens in a tall column is that the pressure increases with the depth in the column of water. And the higher pressure would increase the boiling point. But on top of that, this 58 degrees is only the boiling point of the pure compound, so acetone 100%. And we would never be at this amount. So one has to think of biologically relevant production concentration, so what the cells can still tolerate to produce, and that would be in the range from 1 to 10% maybe. And the evaporation rate decreases linearly with the concentration. In other words, we would be very far from the boiling point. Nonetheless, there is still evaporation just by vapor pressure occurring. And when modeling this, we figured out that this evaporation is enough to keep the concentration of acetone low enough not to be inhibitory to the cells. And that's represented here to the right at the steady state concentration. And the other concentrations are much higher, which means that the cells will not be able to survive. Now, in conclusion, increased temperature favors gas uptake rates and the product to biomass ratio, both very favorable. Now, there is also a limited possibility for volatile product recovery, and acetate and ethanol are metabolically highly favorable. So there are additional solutions needed for other products to be produced, such as co-feeding with other substrates, using split reactors possibly, or other alternatives. Now for the last chapter is called Methylotrophic Acetyl-CoA formation at the origin of life. And so this, as I said in the beginning, is a very fundamental study. And so it's a scientific research question, namely, what is the evolutionary history of the Wood-Ljungdahl pathway? And before I dive into this, I need to give some background. Primarily, I need to explain what the iron-sulfur world theory is. And this is a kind of a theory for the origin of life, as proposed by a man called Günther Westerhäuser in Germany. And he postulates that life emerged at hydrothermal vents. And hydrothermal vents are these like volcanic structures in the bottom of the ocean, where volcanic gases would flow out, which are very rich in interesting compounds. And there is basically a chemical gradient between what's coming out of these spewers and what's in the water surrounding it. Yes, and there, the theory of this iron-sulfur world is kind of rooted in that the chemistry predates the biology. So whatever chemistry was happening there, biology kind of grew around it. And this chemistry would have been an autocatalytic carbon fixation. Which means that it converts C1, inorganic C1 compounds to longer chains, so kind of what we see with acetogenesis. And these reactions would have been catalyzed by inorganic metals, hence the name of this world theory, iron-sulfur, but also nickel, molybdenum, some others. And these minerals are very present in these structures. Now, the other piece of background I need to give is kind of the evolutionary aspects and the term LUCA. LUCA, to define it, it's an abbreviation for the last universal common ancestor. So all the cells all the living organisms that are existing nowadays, they would trace back to one common ancestor, and that common ancestor is not per se one that we found in fossils or something. We would never be able to find it, but it's like it's a thought experiment and a reconstruction of an evolutionary intermediate. And we might know these from phylogenetic trees of, for example, mammalian species or great apes, or etc. There is a common ancestor to that. So if you go far enough back, there's a common ancestor for all life forms. And this would have been dated to 3 or 4 billion years ago. And the earliest division in this last universal common ancestor is the division between bacteria and archaea. And this is represented in this beautiful figure, where bacteria here are to the top, all these this variety of species. And then to the bottom, we have the archaea. And notably, we see that within the clade of archaea, we have also eukaryotes. So we as humans would be somewhere here. And this is kind of the most modern theory of the origin of life, is that archaea gave rise to eukaryotic cells. Because previously they were seen as 3 different groups, but increasingly that's proven incorrect. Yes, and there's certain claims of the Wood-Ljungdahl pathway to be the first of all pathways. So basically, every paper I've read about the Wood-Ljungdahl pathway, and maybe people that are studying this too can confirm, will start in the introduction with, this is the oldest pathway on Earth, and this is what gave rise to life. And there are some compelling arguments to this theory, which is, first of all, that it's spread over archaea and bacteria, so the earliest division. And since it's in both groups, it would trace back to the last universal common ancestor. But also, it's a very simple and versatile pathway, which by that I mean that it's both catabolic and anabolic, so it can form biomass and generate energy, such as these acetogens do. It's also reversible, so often we'll see bacteria that will do it the other way to generate energy. So it's very flexible and versatile. And then lastly, it fits in this iron-sulfur world theory in several ways. First of all, it will use substrates that would have been available in these hydrothermal vents. But also, many of the enzymes involved in the pathway are dependent on cofactors, which are iron, sulfur, nickel, etc. So it looks like a lot like this was the earliest chemistry, but there are also some counter arguments. And the first counter argument is that although it is spread over archaea and bacteria, and it's generally said that they both have this Wood-Ljungdahl pathway, the CO2 reduction from CO2 to CH3 is dependent on completely different reaction steps, different enzymes in archaea and bacteria. And on top of that, it uses different cofactors. So if you recall from the beginning, I was showing this cofactor called tetrahydrofolate, which reduces the CO2. So in archaea, it's a different one called tetrahydromethylotropin. And these cofactors have distinct biosynthesis pathways. So kind of everything points in these being different things. On top of this, the catabolic routes are distinct. So the bacteria, they tend to be acetogenic, so they produce acetate. While on the other hand, the archaea, they produce methane. And that's 2 different ways of generating energy, which do 2 different strategies, which would kind of insinuate that this was not tracing back all the way to LUCA. And lastly, and this is kind of what triggered this study, was the discovery recently of Asgard archaeota, which, well, first of all, have a pretty cool name, but also they're believed to be the ancestors of eukaryotic cells. So that's why they're highly studied. So these Asgard archaeota, like ancient forms of that, have kind of engulfed the bacteria, and that has become the mitochondrion, and that's why we have eukaryotic cells with mitochondria. So that's why these Asgard archaeota are highly studied. But there's another thing that's very interesting in this regard, in the Wood-Ljungdahl pathway aspect, is that we see that these archaea, although they are non-methanogenic, so they cannot produce methane, they still have the Wood-Ljungdahl pathway. So somehow they're kind of in between this classical division of the methanogenic archaea and the acetogenic bacteria. So that triggered this analysis. So the aim of this study was to deconvolute the evolutionary history of the Wood-Ljungdahl pathway and reconstruct its antecedents in LUCA. And I was showing earlier a very complex tree of the tree of life, so to say. And now, just to kind of visualize it in a more schematic way, just to understand what's going on, you could also kind of make a minimal version where we only have 4 species, including 2 archaea to the top, A1 and A2, which have last archaeal common ancestor, LACA, and then the bacteria, B1 and B2, which have a last bacterial common ancestor. And these two then have common ancestor 2, which is the last universal common ancestor. And the same way you can reconstruct these species trees, you can also reconstruct gene trees. And the way evolution works in gene trees is not always that it passes from like vertically from top to bottom. So it's not always Darwinian evolution. It can also be lateral gene transfers. And microbes, they tend to do that a lot, where they'll just pass a gene to another species, and then the gene has it, and it gets a very blurry evolutionary history. And if you have a vertical gene transfer, then the gene tree would look like this, very orderly. It would look a lot like the species tree. However, if you have a lateral gene transfer, then it would look completely different. So the archaeal one here would just be kind of nested within bacterial genes. So looking at a closer look at the phylogenetic trees would allow to tell the origin of that gene, whether it does trace back all the way to LUCA, the last universal common ancestor, or that it occurred much later and only traces back, for example, to the last bacterial common ancestor. So analyzing the Wood-Ljungdahl pathway, represented here to the left, where it's the same figure as in the beginning, but I've added also the archaeal branch here, which looks like a mirror image, right? But it is catalyzed by different enzymes. So we know already from literature that there were some lateral gene transfers, which can be put aside. So we know for sure they don't trace back to the last universal common ancestor. And that is the acetate formation. So specifically the gene ACKA, which produces energy from acetate production. That is specific to bacteria and has been transferred to bacteria, to archaea. And the same way, this H4-MPT, so the right side, the archaeal branch, they are specific to archaea and by accident got crossed over to some bacteria, but do not trace back to them. Then there are 2 modules that could trace back to LUCA, which is the tetrahydrofolate branch, so the CO2 reduction that acetogens use, and there is also this core compound here, so the carbon monoxide dehydrogenase, which puts these 2 molecules together. Certainly what does not trace back is the H4-MPT, so the tetrahydromethanopterin here to the right, the acetate, and the methane production. So since the tetrahydrofolate branch could trace back to LUCA, we needed to dive in a bit deeper to look at every of these genes to identify whether they actually did trace back. And one of the genes that was really telling about this is a gene called FOLD, and FOLD is a key gene in the CO2 reduction, and it's it is without that gene you cannot reduce CO2 basically. And here's the phylogenetic tree from that gene family, and it is a very complex tree. I will give you that, but I have colored the branches so that the cold colors, so green to blue and purple, are showing bacterial genes, and the warm colors, so red and orange, they are showing archaeal genes. So what you see is a primarily bacterial tree, and then nested within, shown by this arrow to the bottom, is a cluster of archaeal genes, which means that the whole gene family has its origin in bacteria, and it crossed over to archaea, which allowed them to use that too. But that means that the last universal common ancestor did not have this gene. So when we go back to the Wood-Ljungdahl pathway, this is basically, these are the steps that the last universal common ancestor had. And there is no way that LUCA was able to reduce CO2. And there's also no way that we was able to generate energy as methanogens or as acetogens. But this raises two questions. First of all, where did this methyl group come from? So by methyl group, I mean the CH3 group that the carbon monoxide dehydrogenase needs. And then the second one is, how did LUCA produce energy? What was LUCA's metabolism, since it was not able to produce acetate or methane? Now, to start with the first question, what is the methyl source of LUCA? We started off by looking into other possible methyl sources by doing a co-occurrence analysis. So we looked at genes that across the tree of life often co-occur with the carbon monoxide dehydrogenase compound, the kind of core William pathway genes. And this way we found a set of genes, methyltransferases, specifically one called MTVC, which co-occurred with this CODH. And this methyltransferase is able to utilize environmental methyl groups. So these are methyl groups that can be bound to different types of compounds. So R just means rests, so it could be anything. And I listed some of them here to the bottom. So methylamines, for example, or sulfur, etc. And these methyl groups, environmental methyl groups, they are basically ubiquitous or highly present in these hydrothermal vents. So I would have had this at this disposition to use. This means that LUCA's anabolism was able to grow from the use of these environmental methyl groups, plus the carbon monoxide, plus this coenzyme A, to produce acetyl-CoA. And acetyl-CoA is the building block for life. So it was able to grow. But this still doesn't say how it got its energy. So it leaves the catabolic question open. Where did LUCA get its energy from? And to answer this, we first need to look at how current day organisms get their energy. And that is in one of two ways. The first way is through phosphoanhydride bonds. So that's shown to the left, and it's a molecule many might be familiar with. It's called ATP. And ATP has these phosphate bonds, which are extremely energy rich. And the breaking of these bonds will release energy for the cells to use in other chemistry. Alternatively, cells can also store energy in membrane potential, so that there's ion gradients within the cell and outside the cell. And these gradients can pump, fuel a pump that also produces ATP. But none of these are present in this ancient William pathway. So there is another theory that we encountered, which is the phosphates. Yes, the phosphate-free thioester world. And in this theory, they postulate that thioesters, so instead of phosphate, we're talking about sulfur, that they have the same properties as these phosphoanhydride bonds of ATP. And one of these molecules that has this sulfur bond is acetyl-CoA. And that is shown here. So this is acetyl-CoA, where, no, sorry, it's only the CoA, not the acetyl-CoA, but it has this sulfur bond here to the end, and this would bind to acetate. And that means that basically the molecule that is produced, acetyl-CoA, is the energy carrier itself. And this was indeed speculated, or that was hypothesized, I should say, by two people called Martin and Tauer already in 2017. That the thioester bond was the early metabolism energy carrier. On top of this, another analysis showed that by Gorter et al. Showed that the phosphate showed a form of phosphate-free proto-metabolism, where they looked at current day metabolism, removed all phosphate, and there was a sort of core which was left, which was enough to kind of sustain chemistry. And most interestingly, you might see here in this coenzyme A, there's two molecules of phosphate. So there is phosphate in coenzyme A. However, in the paper of Gorter et al., they hypothesized that it was not coenzyme A that was playing this role, but its precursor, namely pantothionein, that was the energy carrier. And so here is a coenzyme A, and this is pantothionein. So it lacks these phosphates groups and the nucleotide behind. So this allows us to make a kind of a reconstruction of early metabolism, and also it allows to make a modular representation of the Wohl-Ljungdahl pathway instead of the linear representation we're used to. And this modular representation revolves around two key nodes, which the first one is to the top here, the white one, the CH3 node. So that's the methyl group. And that can be produced from the CO2 reduction, either the archaeobacterial one. Then it can also use from methylotrophy, so CH3 groups, as was demonstrated here. And this can produce methane, or it can go down the carbon monoxide dehydrogenase, so this complex, which is the core of the Wohl-Ljungdahl pathway, to produce acetyl-CoA. And then from acetyl-CoA, which is the second key node, it can produce biomass or biosynthesis as a building block, right? It can produce acetates to regenerate the energy in ATP, or it can just serve as its own as an energy carrier. So then it's this thioester energy carrier. But of course, no single cell has all these pathways, all these modules. So how does it look as a reconstruction of early metabolism? It would look as such, where we see the evolution of it too. And the reconstruction of LUCA, so the last universal common ancestor, is here, where it will it shows in green the catabolic, so the energy generating roots, and in red the anabolic, so the biomass generating roots. So they both come from the environmental methyl groups to produce this thioester energy bonds or biomass. Then somewhere in bacteria, this branch connected, so they were able suddenly to reduce CO2. And by parallel evolution, Archaea did this too, but they defined a different system for that, using a different cofactor. And then further on, it evolved so that these methanogens could then produce methane from the methyl groups, and the acetogens could produce acetates. So this is how one original metabolism kind of evolved in these distinct pathways. Now, in this paper, or in this manuscript, we revised the theory for the origin of life, where CO2 reduction was not at the origin of life. It could not have been because the genes were not present. And we proposed that the methylotrophic acetyl-CoA pathway was, and that is both an anabolic and a catabolic pathway, and we show how, that it fits in the iron-sulfur world theory, but also that it fits in this phosphate-free thioester world theory. So in conclusion, the methylotrophic acetyl-CoA pathway fits all the criteria to be the first metabolic pathway predating life. So to conclude, chapter 1, we saw that acetogenesis can be a renewable alternative to olefin production. In chapter 2, we identified the gas fermentation benefits of high temperature, but that's diversifying the products will remain a challenge. And in chapter 3, we hypothesized that the methylotrophic acetyl-CoA pathway fits all the criteria to be the pathway that enabled life. So yes, I would like to acknowledge some people. First of all, my supervisors, of course, Alex and Torbjørn. Also, not here today, but Professor Alfred M. Spormann, who was supervising me at Stanford for the last study. Then my examiners, thank you for taking the time to come here, also traveling from far. So Kasper, Pablo, and Niels. The coordinators that made all this possible. Also my lab. Thank you. It's quite a list, so I'll not name everyone. The lab in the US. Also, generally, all PhD students at CFB and all staff for being a great working environment. I would like to acknowledge also the funding agencies. So Novo Nordisk Foundation that paid my PhD, and also Otto Mønsted, Christian and Ottilia Brorsons Rejselegater, and William Demant Foundation that financed my travels. Yes, thank you.